Welcome to G Fun Facts Online. Today, we're talking about a technology that sounds like it was lifted directly from a science fiction novel. What if I told you it is now medically possible for a child to be born with genetic material from 3 different people? This isn't a story about the future. It's happening right now. It's a revolutionary procedure that offers hope to families facing devastating genetic diseases. But it also opens a Pandora's box of ethical questions that we are only just beginning to answer. This is the story of 3-person IVF. To understand why this technology is so revolutionary, we have to journey deep inside our own cells. Think of your body as a massive city, and each of your trillions of cells is a building. Inside every building are thousands of tiny power plants. These are your mitochondria. Their job is to take the food you eat and the air you breathe and convert it into energy, the fuel that powers every single thing you do. They are essential for life. What makes mitochondria unique is that they have their own tiny set of instructions, their own DNA, completely separate from the main genetic blueprint stored in the cell's nucleus. This mitochondrial DNA, or mtDNA, contains just 37 genes. But here's the crucial part. While the nuclear DNA in the nucleus comes from both your mother and father, your mitochondrial DNA comes exclusively from your mother. It's passed down through her egg cell. This is where the problem starts. If a woman's mtDNA contains harmful mutations, she will pass those mutations directly to all of her children. And because mitochondria power every part of the body, these diseases can be brutal and widespread. The organs that need the most energy, like the brain, heart, and muscles, are often hit the hardest. The diseases are horrific. Take Lee syndrome, which usually appears in a baby's first year. It causes a rapid loss of mental and motor skills. Infants can lose the ability to eat, suffer from constant seizures, and the prognosis is tragically poor. Then there's Miller syndrome, which often strikes in childhood, causing stroke-like episodes that lead to brain damage, vision loss, and dementia. For families, it's a nightmare. And for mothers who carry these mutations, the choice to have children is terrifying. They face a genetic lottery. A mother might have no symptoms herself because she has a low percentage of mutated mtDNA. This is called heteroplasmy, a mix of healthy and faulty mitochondria. But when her eggs form, a process called the mitochondrial bottleneck means that, by pure chance, some eggs can end up with a devastatingly high load of the faulty DNA. A healthy mother can have a severely ill child, and there's no way to predict it. It's this roll of the genetic dice that created the urgent need for a new kind of solution, one that could sever this chain of inheritance entirely. So if you can't fix the faulty power plants, what do? You replace them. That's the elegant and incredibly complex idea behind what's properly called mitochondrial replacement therapy, or MRT. The goal is to create an embryo that has the nuclear DNA from the mother and father, which determines all their personal traits, but with healthy mitochondria from a third person, an egg donor. There are 2 main ways to do this. The first is called maternal spindle transfer, or MST. This happens before fertilization. Scientists take the egg from the intended mother, the one with the faulty mitochondria. They carefully remove its nucleus, which is held in a structure called the spindle. They then take a healthy egg from a donor and remove its nucleus, leaving behind the egg cell full of healthy mitochondria. They then perform the transplant. The mother's nucleus is placed into the donor's empty egg. This reconstructed egg, with the mother's DNA and the donor's healthy mitochondria, is then fertilized by the father's sperm. The second method is called pronuclear transfer, or PNT. This is the technique pioneered in the United Kingdom, and it happens just after fertilization. Here, you start by fertilizing both the mother's egg and a donor's egg with the father's sperm. A few hours later, you have 2 very early embryos. The mother's embryo has the right parental DNA but faulty mitochondria. The donor's embryo has healthy mitochondria but the wrong nuclear DNA, Scientists then remove the pronuclei, the little packages of DNA, from the mother and father, from the mother's embryo, and transfer them into the donor embryo, which has had its own pronuclei removed. The result is the same: an embryo with the nuclear DNA from its parents and the mitochondrial DNA from a donor. Both of these techniques are incredible feats of microscopic surgery. And they are working. This isn't just a theory in a lab. The United Kingdom became the first country in the world to formally legalize MRT back in 2015, but only under very strict regulations. The program at Newcastle University has been the global leader. In a landmark report, they announced that several healthy babies have been born to women at high risk of passing on severe mitochondrial disease. The parents' relief is palpable. One anonymous mother said, "After years of uncertainty, this treatment gave us hope. And then it gave us our baby. We look at them now, full of life and possibility, and we're overwhelmed with gratitude." This success is the result of decades of careful research and public debate. But the UK's cautious approach is not the global standard. The very first baby born using MRT was in 2016 to a family who traveled to Mexico, where a US-based team performed the procedure to get around the effective ban on this research in the United States. Other births have been reported in Greece and Ukraine, sometimes not to prevent disease, but as a controversial treatment for infertility. This has created a global patchwork of laws, where access to this technology depends entirely on where you live. Naturally, a procedure that creates a baby with DNA from 3 people sets off a lot of ethical alarm bells. Let's tackle the biggest ones. First, is this a 3-parent baby? While it's a catchy headline, most scientists and ethicists say it's misleading. The donor contributes about 0.1% of the child's total DNA, just those 37 mitochondrial genes. The other 99.9%, the DNA that codes for personality, appearance, and intellect, comes from the intended parents. UK law is clear. The donor is anonymous and has no legal rights or responsibilities as a parent. They are treated like an organ donor. The more serious debate is about germline modification. This means we are making a genetic change to an embryo that can be passed down to all future generations. For decades, this was considered a line science should not cross, for fear of a slippery slope towards so-called designer babies, where we might try to edit genes for intelligence or athletic ability. MRT is, without question, a form of germline modification. The healthy donor mitochondria will be passed on if the child is a girl. Critics argue this opens a Pandora's box. Proponents, however, say MRT is a unique and special case. It doesn't alter the core nuclear DNA. It replaces a whole, naturally occurring component to prevent a horrific disease. The UK regulations have built a legal firewall, restricting its use only to preventing serious illness. The question is whether that firewall will hold. Then there is the moral status of the embryo itself. The pronuclear transfer method is especially controversial because it requires creating and then destroying an embryo to harvest its healthy mitochondria. For those who believe that a human life with full rights begins at conception, this is a grave ethical violation, treating human life as a means to an end. Others argue that at this very early stage, the embryo doesn't yet have the full moral status of a person, and the immense benefit of preventing a fatal disease outweighs the loss. This is a fundamental disagreement that may never be resolved. As with any pioneering technology, there are also risks and unknowns. The biggest biological concern is something called mitochondrial carryover. It's impossible to perform the transplant with 100% purity. A tiny fraction of the mother's faulty mitochondria can get carried over into the new egg. The hope is that this amount is too low to cause disease. But in some cases, scientists have seen a worrying phenomenon called reversion, where that small, carried-over population of faulty mitochondria starts to outcompete and multiply faster than the donor's healthy ones, potentially increasing the risk of disease as the child develops. This is why lifelong, careful monitoring of all children born through MRT is absolutely essential. We are navigating uncharted territory. And the story doesn't end with preventing disease. The technology is already being used for a much more common and controversial purpose, treating age-related infertility. The theory is that older eggs have less efficient mitochondria, and that swapping them out could recharge the egg. Clinics in countries with lax regulations are already offering this, moving the technology from preventing death and disability to treating a natural biological process. This is the slippery slope that critics have long feared, and it's a debate that is only going to intensify. The dawn of three-person IVF is a pivotal moment in human history. It is a technology born from the profound desire of parents to give their children a healthy life. For a small number of families, it has been a miracle. But it has also handed us the power to edit our own biological inheritance. It forces us to confront what it means to be a parent and what limits we should place on science. The healthy babies born so far are a testament to scientific genius, but their existence reminds us of our immense responsibility. How we choose to use this power, the laws we make, the ethical lines we draw, will define the legacy of this genetic revolution for all generations to come.